let me begin with a simple observation. We all start from a single cell. And from that cell, a fully functioning and structured body forms with extreme reliability. That's essentially what development does. And what makes this remarkable and surprising is that the molecular processes that are driving development are noisy and stochastic. And so the question I want to address today is simply how living systems convert this microscopic fluctuations into macroscopic reliability. Because when you look at physical systems that are far from equilibrium, like snowflakes, the formation of snowflakes, well, no two outcomes are identical. It's chaotic almost, right? However, living systems are different. We all have 5 fingers. We rarely see people with 6 or with 4, so it seems to work quite reliably and reproducibly. And of course, you all know the example of identical twins and how difficult it is for us to distinguish between them, right? And so reproducibility is definitely not automatic. Development must actively control that. And that means that the system must be constrained. Not all microscopic fluctuations are allowed to propagate into macroscopic outcomes. That's what a constraint basically means. But constraints, they must also leave quantitative signatures. And so if development is truly constrained, we should be able to measure those constraints quantitatively. And so the real question then becomes, how precise is development numerically? And so let's measure this in 3-hour-old fly embryos, which is my favorite system of investigation. Patterning happens very quickly. The genetics is extremely well defined, and you can start doing quantitative measurements. And so we ask a very simple experimental question, basically, how precise are the molecular readouts that define pattern boundaries? Like, for instance, this one here in red, that splits the future head from the rest of the body. And once you formulate the question this way, it becomes measurable. So here you see roughly 100 fly embryos. Well, not yet. I see them. Now you see roughly 100 fly embryos. So up here is the embryo from the previous slide. And if you section it, if you take cross section of this, you get basically this. And from the back of the room, certainly it already looks very similar, reproducible. But you can, of course, quantify this statistically. And then with the help of computers, and you see something striking, it turns out that the boundary position, varies by only about 1% of embryo length. And that corresponds to roughly 1 cell diameter. All right, so at 3 hours, we basically have a positional position that is at the scale of the smallest spatial unit available to the system. But we can go, so that's already remarkable, right? Going back to the identical twins, it comes a little bit more understandable why it's so hard to distinguish them, because development actually works even with these wild type embryos quite remarkably reproducible.
You said 3 hours. So
in that it's a good question. I've never measured how precise it is in time. Yes, good point. Now we can go one step further. Actually, I've never looked at temporal precision. Experimental conditions need to be very stable, etc. So temperature, all has an influence, of course. So it's yeah, maybe in my next career. So now we want to go one step further, and instead of measuring morphological boundaries, we can actually measure the positional precision directly from the underlying molecular morphogen gradients. And there again, the numbers are very strong. So here you see the underlying gene regulatory cascade. Yes. Yes, there it is. It's basically an interacting network of cross-regulating transcription factors. They are initially broad molecular gradients that progressively get refined into sharp expression boundaries, one of which corresponds exactly to the morphological boundary that you saw on the previous slide that we just measured. And when we measure the concentration patterns across many embryos, the molecular levels vary by about 10%. So this fluctuation here is about 10%. And that is already remarkably reproducible at the molecular scale. But because these gradients are steep, simple error propagation tells us that this level of molecular reproducibility translates to about 1% in positional precision. Okay, here these peaks or even these boundaries. And that's, again, roughly 1 cell diameter. And so development operates very close to the physical precision limit that's set by molecular noise and cell size, at least at this stage of development. And that means that reproducibility of 10% is also sufficient to generate basically near single cell positional precision. Okay, so that's an important take home already. But of course, precision at the boundary is a local statement, and we get an even stronger constraint when we look at scaling with system size. So now on the next slide here, I show you 3 embryos from different species that show that the patterns don't just appear at the right place, but they appear at the right relative place. If the embryo size changes, even by a factor of 5 here across different species, the patterns here of that same gene are positioned proportionately to the size of the system. Okay, and we can actually quantify this if you just look at Drosophila, which is the guy here in the middle. There you see that in the natural fluctuation of a New Jersey population of fruit flies varies in size by roughly 10%. But when we measure the gene expression pattern and rescale and plot them rescaled between 0 and 100% egg lengths, well, the pattern boundaries or the peaks here, they fluctuate by only 1%, which means that the cells locally must know where they are in proportion to the size of the system. Basically, the local behavior reflects global geometry. Okay, yes?
Bigger embryos and also bigger cells or more cells?
Very good point. They have actually, between the different species, they have bigger cells. They have exactly the same number of cells. Between different Drosophila, I have to measure. Sorry. I would need to, yes.
Positive forms for the cellular regression. Is it fair to say that since these gradients and expressions exist well before even cellular regression is happening, and so when the cells basically take up a certain amount of, basically from the membranes, are they doing some sort of binning? Each cell is just basically taking everything in its region and summing it up, which reduces the noise.
Right, yeah, that's a very good point. Exactly. So we're going to get to this. There's, of course, noise, molecular noise in the system. And one of the ways of dealing with how do we make sure things work in the single cell level, yes, is that groups of cells take decisions together. And one of the way of doing this is, in fact, yes, by signals that are sent out by one cell and measured collectively by a number of cells, and spatial and temporal averaging is at place. That's absolutely true. Not just in space, but also in time. Like, for instance, for one of the gene products, the mRNA we measured has a lifetime of an hour. So they hang out for a long, long, long time. So that helps a lot with the averaging, but that is still not enough. You need also a certain amount of temporal averaging in order for things to happen. Yeah. It is true. Now we can, so you give me a good idea, because I know all of the measurements I show you today, they were actually done in 2D. So 25 years of my career 2D measurements, and right now finally I have a student in the lab who managed to get 3D to work. So we have now all of the nuclei, and one of the first things we can now address is exactly that. We don't even need to stain, right? Can just now look at and count. I will send him a text just after the talk.
Question, because it seems that the precision is up to the size of the cell each time.
Yes.
So do you understand why? Because when you have diffusion of molecule, for example, so I understand that this is a cell that takes this decision. So, but you could have subcellular precision in a gradient, for example. So I was wondering
Yes.
whether you see how you explain this relationship
Right.
between the cell size and
Right.
the precision that you get.
Good point. And I give you my hand-wavy physics kind of explanation. The smallest physical unit that can make a decision in the system is a cell. It's a cell in the end that will decide whether or not to belong to a pattern, or whether or not to belong to that pattern or to the pattern on the other side of the boundary. That is the decision maker. And so I think evolution evolved the precision, if you want, to what's necessary. And there's no point of going further. It just costs more for nothing, right? Because there's not really anything we can gain from it. It turns out there is overlap because, like, if you look at the boundaries, they're not exactly straight. They're slightly jagged. And for that, to cope with that jaggedness, you need better than single cell precision. But it actually turns out I'm lying. The precision is actually not at a single cell. If you really look carefully, the precision is at a half of a cell. And that half of a cell is just enough to get rid of the jaggedness. But in order to get to the precision of a thing, here I'm looking at single genes, right? I'm showing you single genes, or I'm showing you this big boundary. In order to get to the precision of half a cell, you need to look at the precision that is generated collectively by the network of genes. So you need to construct a covariance matrix, normalize, and then you get to have I mean, I don't know if I was clear, but it's only when multiple genes act together that you really get to have all right? But this discussion shows you already how far we were able to push quantitatively into the system. That we have access to very minute questions like those. Right? All right, so what I said is that local behavior reflects global geometry, and that can really only happen if that cannot happen if the regulation would be purely local, and this implies there must be global constraints present in the system. All right, and so experimentally we now have three very strong observations or signatures, let's call them. There's reproducibility, there's precision, and there's scaling. All right, and so to recapitulate, basically what development does here is to link molecular fluctuations, transcription factors essentially binding to enhancers, all the way to organism level structure, right? And transcriptional dynamics, basically network dynamics, as we just discussed, influences cell fate. Cell fate decisions shape spatial patterns, and the patterns then give rise to tissues and body structure. And so variability at the molecular scale is then propagated and filtered somehow across multiple levels of organizations, both in space and time, across many decades of scales. And so in physics, when we see systems that have these properties, reproducibility, precision, and scaling properly, we usually suspect constraints. Okay? Basically principles that shape how variability can propagate through across different scales. That's what constraints here mean. right? They don't mean detailed mechanisms. In biology, detailed molecular mechanisms. They basically simply only mean these macroscopic signatures for constraints. They really only mean that not all microscopic trajectories, as I said, produce viable macroscopic outcomes. Okay, and so what I call constraints at the macroscopic level, so producibility, precision, and scaling, they should really just be macroscopic signatures that reflect constraints at the microscopic level. And the question that I want to ask next is then whether we can see that really in gene regulation. Do we see microscopic signatures of such macroscopic constraints in the underlying molecular constituents of transcriptional regulation? Yes, sorry?
Because this is very transcription centric, but what do you do with signaling, for instance? Is that part?
I don't do much with signaling, but I suspect something similar you would see.
In the beginning, it's not critical. But no.
Right. Exactly. So I tell you now why I use transcription, but I suspect for signaling something similar is at play. But for signaling, I have absolutely neither the macroscopic nor the microscopic insights because I've never really looked at it. But because again, it's molecular and it has evolved, etc. Pathways are constraints, or pathways are not constrained, conserved. Across many different hierarchies and levels of biology. Like transcription, I suspect for that reason something similar is at place. But one would have to hunt and find this. Okay, I give you a very conceptual introduction into how things were built in my lab over the years, and I only see this now. Putting all of this together the way I'm trying to put it together right now would have been difficult 20 years ago. 20 years ago, you jump into a system, oh, you see reproducibility, oh, you see precision, oh, you see scaling. But what does it really mean? Well, today I ask, does it mean that they are signatures of microscopic constraints? Okay, so do you understand all the question? Ask me, this is very conceptual, but I kind of like to give you a conceptual, you'll get something molecular in just a few slides. Don't worry, yes?
Is there any evidence that a certain amount of fluctuations is necessary for development to proceed correctly? As in, if you could somehow reduce the fluctuations to zero, you would also not end up with proper development.
Yeah, that's kind of what we think about evolution, right? You need it for evolution, and so that's what I reflect back to you. In development, I don't know. But for evolution, obviously, yes, it's necessary.
Can you envision a developmental system which is precise but not reproducible? I was wondering what is. I think I understand the meaning of the difference between reproducibility and precision that you measure. But for me, they are very intertwined.
Absolutely, they're totally very. And to go one step further, we even showed that scaling and precision are hugely intertwined. I'm not going to go. This is a big digression, but scaling and precision are very intertwined too. In fact, I showed you, right? 10% fluctuation in size goes to 1% precision, and that gives you a signature that they must be connected, right? The difference conceptually between reproducibility, or how we see it, between reproducibility and precision is of course that reproducibility really is across species, across samples, individuals, and precision really is within. The boundary is jagged, and it's this nucleus and not this nucleus that expresses the pattern. That's really precision, and that precision happens to be at a single cell level. Yes.
Sorry, I'm having a confusing thought, but you can talk about precision just when you compare things, right? So the precision is one, but you know that it's that precise because you compare different individuals, and it's always that one cell.
The way I constructed it in this talk, yes.
Okay.
I don't need to do that. I can also look just at individuals, but still use several individuals for statistics. I'm allowed to do this.
Yeah.
Right? You need statistics.
The concept of precision comes from, right? Because if you only have one
embryo
that you're looking at, how do you define this as the precision of the pattern?
Well, great, but it basically that this embryo knows I'm sitting, because embryos don't talk to their friends, but they still know I sit at 34% and make a cephalic furrow and not at 35% and make a cephalic furrow.
All right.
That is precision because it happens within an individual, right? Reproducibility is that different mothers, or even the same mother, produces an egg such that it always makes a cephalic furrow at the same position, because the mother puts goodies in the egg before development starts, and she can do that with utmost reproducibility because she reproduces the same egg over and over, right? So is there a time cap for me? Because I'm with all your questions, it's great, but I don't know how much I get done in my talk.
I think we can do until 10:30.
Okay.
Still buffer that time with the.
At some time you will need to give me space to talk. Well, or not. I don't need to finish actually. All right. So where were we, right? So can we see basically this in gene regulation? And so basically, are there quantities in this system that remain stable across space, time, genes, or perturbations? Okay, and that's what physicists would call invariants or laws. So we are trying to go now at the microscopic level of transcription. We are trying to identify parameters that describe transcription. And then the question is, are there relationships between these parameters that look like empirical laws that we know from physics, or even relationships that are just constant or invariant, right? And historically, this is exactly how empirical laws have been discovered in other complex dynamical systems. Like, for instance, in planetary motion, right? Kepler found empirical regularities long without knowing any mechanism, and long before Newton explained them by gravitational dynamics, right? And so we ask the same question in development, basically. Do stable, measurable relationships govern gene activity? That independent of the specific gene or the regulatory context, okay? All right, so if we want to do this, we need to test this, you need calibrated real-time measurements. And you get this in the fly embryo by looking at transcription. That's a very good way to get in. And so it's one of the few places where gene regulation basically becomes a direct observable in time. And it's also the important mechanism that determines cell fate and then patterns and then structure. I mean, it's the thing that links, in a way, all of the scales that I talked to you about before, that we want to cross, and that these constraints are signatures for, right? And so that's essentially why we focus on transcription. And the fly embryo is beautiful because you can basically measure simultaneously transcription at a single locus and observe the full pattern as you saw it. So these are individual transcription spots here. Those are nuclei, and here you see the 7-stride pattern as it forms life, okay? And so basically now using this quantitative imaging and a little bit of modeling, we can effectively turn this embryo into a laboratory and measure gene activity at multiple scales simultaneously. And all right, and so let's begin with the macroscopic level, the macroscopic patterns. And this is something we knew before. So basically these are reconstructed gene expression patterns for 4 genes that we measured simultaneously from antibody stainings. And what you already see here is that the macroscopic dynamics of these patterns is highly complex and dynamic, even though they're all regulated differently at different spatial and temporal locations. Okay? But if these invariant relationships exist, they must be present in the dynamics themselves, in the underlying transcription dynamics, and not just in the averages here. The averages, again, tell us, well, there's a signature that we need to find something. Okay, and so that means we need to really go at a single locus and check there. What happens really at a single locus that is able to produce these highly complex but reproducible dynamics. Okay, it's a very beautiful, highly orchestrated dance of these four genes that work together. And now we're zooming in onto a single locus, and all the mess comes to light. So here on the little zoom in, you see a single locus, very bright. The guy who's sitting in the last row there made that movie, former grad student in the lab. Here's the single locus, very bright. There's many mRNAs at once. We label individual mRNAs, and you see them come off, diffuse off this single locus. Okay. And at the same time, if we zoom out, you see the single loci in green here forming a pattern. This is just a pattern boundary that will form roughly here in individual nuclei. Okay, it's a technology that took us roughly 15 years to develop. Don't worry, the graduate student didn't stay 15 years in the lab, but it's many generations of graduate students and postdocs. It's not easy to get there. Each right spot corresponds to a single transcription site and a single nucleus. And if you want, that is the microscopic origin of variability. This is where variability is born in the system, okay? At least for the question we want to ask today. And the question is whether this variability is unconstrained or whether it obeys some empirical rules. And to answer that, we need to really quantify of what we're observing here. And so from these kind of movies, we then reconstruct transcription activity in absolute units. So here you see we're measuring in many embryos. Let's look at one spatial location. Here's an example from a movie. And at one spatial location, we have many nuclei, many loci. And here you see now the transcription activity as a function of time. It looks like a mess. Very, very noisy. As it should, because it's a molecular process. It's a stochastic molecular process. You see that here, here we have a mitotic event. So there's one interface, mitosis, another interface. So transcription shuts down completely. You see how our noise, here's one nucleus that doesn't show anything. That's how low the noise level is. And now now instead of looking at isolated loci, we can try to reconstruct the full pattern again. And so now let's focus on just one particular time point here. Okay, and average over time for this spatial location, and do this for all spatial locations marching along the embryo. And what you see is we recover the patterns that we saw previously at the protein level. Okay, but now they were constructed, of course, from an individual locus. You already see they're highly structured across the entire embryo. It's not random. They're highly organized. And what's crucial here is that these measurements are calibrated in absolute units by matching basically these live measurements that I showed you before from completely independent measurements that are done with single molecule fish. So where we count individual mRNA molecules, again, in those four genes. Okay, so there's at that very same time point. So this time point, I choose this time point for a reason, because at that time point, we have fixed hundreds of embryos, SM fished them for those four genes, absolute measurements. We have those same measurements live. And now with a single conversion factor, we can morph one data set on top of the other. And that conversion factor gives you mRNAs per minute, an absolute unit. Okay? Yes.
Standard error.
Pardon?
How is the error? Because here, this is a mean that you show, or there is the error with small, there is no error bar.
There is error bar.
So this is a tiny thing.
These are, yeah, exactly. It's good.
Yeah, it's good, so good, like, I see it.
Exactly.
Oh, yeah, I'm here, so you see.
That's why in the first row, yes. Forward, because you would have thought I lie when you sit in the last row. And so absolute units are important, of course, because they allow us to make a quantitative comparison between genes, different embryos, perturbations, and all this good stuff that's about to come, okay? And that's basically also essential if you want to test for generality. And so absolute units are really, really important. They're also important for the modeling, but that's an aside. Now, yes, sorry.
This might just be a detail question, but you said that you could relate the abundance of the mRNA from the fish to the live transcription rate.
Yes.
Does that mean that all the genes have the same degradation rate?
That's one of the assumptions, yes.
Okay. Just because the ratio of the two would give the...
Yes,
okay.
exactly. That's it. But that's one of the assumptions.
Okay.
And also the same elongation rate. Right. Good point. But it's not, well, we can talk about it later. If you have issues with it, it's not such a far-fetched assumption. All right. So obviously the patterns are not static. They're built dynamically in time, and because we have temporal now, we can animate them again. It's not, no, sorry, this is not animate. This is the measurement now in time, right? Previously, the protein stainings, they were animated because they were reconstructions from many, many embryos that we fixed. And we took cohorts at different 5-minute intervals. Here, this is the direct dynamics that we measure directly. But we reconstructed now from single loci. And you see how similar to the actual protein dynamics that they look. But now reconstructed from the single locus dynamics that looked like an absolute mess when you look at it as a function of time. In the top level, you see what we actually measure, averaged across hundreds of loci, the same position along the whole anterior posterior axis. And in the bottom, you see, because we have absolute units, you see mRNA per minute, so the rate. One anecdote, this line here shows you basically the peak, and note that all of the genes roughly go into this peak, which is kind of surprising, right? So it means that the fully on state of all of the genes is 15 mRNAs per minute. All right. But as I said, so they're highly structured, right? So meaning it's orchestrated, there's another microscopic constraint if you want at this level at least. But that structure must somehow already be seen in the dynamics at individual loci. Okay, at least that was our hypothesis, and that's what we wanted to see. And so now let's go to the elementary unit, the single transcription locus, and see and view this as a basically stochastic process evolving in time. All right. And so when you look at the single locus over time, transcription, of course, doesn't occur smoothly. It occurs in so-called bursts. There are regions where transcription is on, and there are regions where transcription is off. Right? When it's on, polymerases can bind and mRNA is produced. And a minimal way to describe this is by a small so-called switching model. Okay, it's a minimal model if you want, where you have periods where the promoter is in an on state and periods where promoter is in an off state. And from this, you can now extract a small set of physically interpretable parameters. Basically, how often does a burst occur? How long do they last? And how much mRNA is produced when the promoter is in an on state? All right. And then from these very intuitive parameters, if you want, you can now construct composite quantities. For example, the transcription rate R, because that's important, because that's the rate essentially that governs how much is the locus producing. That's really the thing that we need to get at. But you can also have something that we call the probability of a locus of being on, P on here, okay? Or the switching time scale that the locus switches between on and off states. Okay, I didn't put the formula for this in because in being respectful for the biologists in the audience, I didn't want to overload the slide, but there is also this quantity of switching time scale between the on and off state. Yes.
But the T on, T off are distributions, and this is the mean.
Excuse me?
The T on, T off are drawn from some distribution. And what you use here is the mean, or you use fixed value.
So, good point. But in the next slide, you'll see. So this here is just give you the model. You'll see the distribution and ask the question again if it's still unclear. So here it's really a single trace from which we extract the corresponding T on and the corresponding T offs. We can do this from single trace. This is why it's a microscopic measurement. Okay, but we need to convert somehow the activity that we measure in the upper panel into individual binding events for polymerases, which are the gray lines. And I'll tell you in the next slide how we do this. But let me finish this here. And so there's now different ways how these individual parameters could control the transcription rate. How they could modulate them. One is the initiation rate K. Yes, how many mRNAs do we produce, or how many polymerases are loading in one on event, or by modulating the distance between two bursts or the length of a given burst. Okay, all of these parameters allow us to modulate transcription. And the question is then really, can we measure these parameters from data? Okay, so this is just the little model that in which we operate. And so now you're going to see a single transcription, a real measurement here. So that's how traces look. You see the bursts already in the trace, right? But in order to really analyze what's going on, we need the individual initiation events. All right? But because each mRNA is labeled and remains on the locus for a certain amount of time, the time it elongates, we need to back that out. Basically, we need a little model for transcription elongation, and then reconstruct computationally the trace of polymerases that come in. And we use a little inference approach. I can go into the details. I want to skip it. Basically, so basically now from these inferred initiation events, we can then estimate the instantaneous transcription rate at the single cell level, which you see down here. So here we have now mRNAs per minute reconstructed from this trace up there. And this is again so this is now the rate for a single locus. It's no longer the average that I showed you before. This is the rate at a single locus. All right. And now this rate we can segregate in on regions and off regions, off periods and on periods, which identifies now individual transcription bursts. And so now for each locus in each nucleus, yes, sorry?
Strictly speaking, do you need the elongation rate? Because I was thinking that if you just have each of the microscopic beginnings of signal when it's in the on state, however long it takes, doesn't necessarily matter.
No, so sorry, I wasn't exactly clear. Sorry, I just need to order my.
That's the detail that you can just.
Well, it comes from the fact that it's the mRNA that's labeled. It binds to the locus, and hangs out there. But you kind of need to back that out. It's like as if, you know, what you see is the convoluted fluorescence.
lingers
Exactly,
beyond
it lingers around.
determination
Exactly,
functions.
exactly. And so
You have to, it's a pile.
it's like a cake kernel that you need to take out. Okay, and
Thank
for
you.
that kernel, you need a model. All right. All right, and so for each trace, for each locus, we get now an individual trace. We can back out the rate and we can back out the on and off periods. And so for here, 200 nuclei, basically for this particular gene, we have these matrices, if you want. And from those, you can now, instead of looking at an individual trace, we can now analyze the statistical structure of these parameters across space, time, and later also genes. Does that answer your question? And sorry, yeah, yours I have, I guess, and yours about the statistics, right? So each individual locus gives us now one line in these matrices here. So the analysis itself is technical, but conceptually it's really simple. From single locus dynamics, we can now extract this small set of physically interpretable parameters across these thousands of loci. Let me do this for one in particular. So this is the rate as a function of this probability of being in the on state. Okay, on the x-axis. So you have 0, the gene is fully off. You have 1, the gene is fully on, and on the x-axis, on the y-axis, you have the rate. And so if these were independent and independently regulated, you would expect data points to be all over the place in this space. But it turns out they all collapse to a very simple curve. All right? And across space and time, then, R is almost entirely controlled by this P-on control knob. And so the apparent complexity of all of this bursting seems to reduce here to a very low-dimensional relationship. All right? Now, this is just a relationship between P-on and R. If you look at the other parameters, it turns out all of them collapse into very low-dimensional relationships if you look at them as a function of P-on. Yes?
Be on and off, you can still have the same fraction of the time on, but you could have a very different rates of switching between on and off, right? And that can basically tell you like the gaps between the burst and off.
So the rate of basically there's a switching, there's a characteristic time scale with which you switch on and off, and that we can measure. And in fact, it turns out that this here, this TC, the last one, is that characteristic time scale. But look at how it varies as a function of P-on. How would you say it varies? It doesn't vary, it's almost constant. And this is just one data set for now. These parameters are all not independently regulated. They all kind of contained in these low-dimensional relationships. And what's even more striking, this is true for all of the genes that we looked at. Damn it. Okay. And so these relationships basically occur for all of the gap genes, despite them being deregulated completely independently. All of their regulation has evolved completely independently. They have all different enhancers, multiple enhancers that operate differently in time and space, and yet, boom, they all fall on the same relationships.
Does it compare to something?
Pardon?
Does it come?
Hang on. It all falls apart. That's why I'm giving this talk. All right, so in practical terms, there's basically two relationships that are approximately constant, this K here and this TC. The initiation rate and the switching correlation time are approximately constant, okay? And now the next thing is that we would like to test this by perturbing the system. And so we perturb the system by, for instance, taking out an entire transcription factor. In the flies, this is easy. You take a null mutant, you take out a transcription factor that regulates or that is within the network. Very complicated. Or you take out an enhancer of an individual gene. Or you change the promoter architecture. Whichever we try to do, we manage to get a very strong effect. Here you see the difference between wild type and this deletion of this one gene. If you look at output, they fall again on the same relationships. In gray, you see the previous one of the gap genes, of the wild type. All right? And if you put all of our data sets together, but this is now over 30,000 data points when it comes up ever here. So it looks now, of course, more cleaned up because we have a lot of data. 30,000 data points, they all collapse. And because this T, C, and K here on the right, because they are approximately constant, the transcription rate R is largely determined just by this P on. And the on and off times are also tightly constrained as well. They're basically giving you antisymmetric relationships. Okay? And note that there's a single control parameter. Everything collapses to a single control meter, this probability of the locus being on or off. And from there, everything is determined.
Is it then fair to say that regardless of the gene, when it is on, it produces the same number of mRNA per minute, regardless of the length of the mRNA?
Yes. Yes.
But they might take a slightly different time to finish them because they are of different lengths. But they initiate at least.
Yes, right. We are looking at the initiation events, and those are the same across all of these genes. And so basically, transcription regulation operates in an extremely low-dimensional control space. And these parameters are now examples for those microscopic constraints that we were basically looking at that could explain the macroscopic signatures, the producer position and scaling that we saw earlier, okay? And now the next question is then whether the structure is specific to these particular fly genes, or if we can also see them differently from the gap gene network. And now I'm showing you data that comes from different labs, because I didn't go in and develop a research program. So it's difficult to find in different labs because you need similar kind of measurements. Many things change, the analysis, the imaging, the processing. And so this all has to be taken with a grain of salt. But nevertheless, again, they fall on top of our relationships. You see, there's a slight change. So this is another eukaryote. This is a yeast gene here. There's a slight change in these absolute units, but it's always much more complicated to get absolute units to work out between different labs, different experimental setups than relative or than these functional relative relationships. But you probably agree that the functional relationships approximately stay the same, right? Yes?
Can you just repeat what K is again?
K is the initiation rate. Once the gene is on, what's the rate of polymerase binding? Yeah. And finally, when we look at endogenous genes in human cells, we again observe the same relationships, even though now the absolute changes are larger. But again, it's difficult to compare things across different labs, right? And so taken together, basically these results suggest a quite coherent picture. First, the transcriptional output is regulated just primarily by this one control parameter P on. Second, the characteristic timescale TC remains strongly constrained. It's basically quasi-constant or quasi-invariant. Invariant like energy in physics or angular momentum for Kepler when he looked at planetary motion. And as a consequence, it turns out that transcriptional dynamics seems to follow very low dimensional rules. Okay. And these relationships, I hope to have convinced you that they're robust against genetic perturbations. They appear conserved across a different eukaryotic systems. And at least at the level of transcription dynamics, development begins to look like a system that's governed by maybe empirical physical constraints. Okay. And now the natural question then becomes whether these constraints leave also measurable signatures in other developmental systems.
Right, because I don't get mad down, but I think the necessary or sufficient condition to get precision and reproducibility. That's what you're observing.
So currently, the precision, reproducibility, and scaling, I sell them to you as signatures for why did I go looks like microscopically.
Yeah.
The causal link, I still need to do.
Don't know whether this is sufficient or necessary to get.
I don't know yet. But I used it as it smells like there must be something in the microscopic dynamics. So let's look. And in fact, we find something. Okay, I would be surprised if they're not somehow connected. But you now need to come up with perturbations that directly affect K, T on or T off in one way or another. And then see whether now when I can perturb, when I manage to, because all the perturbations I tried so far, they are upstream of those three parameters and they don't do nothing. Which is a strong indication that these are universal. But now I can now think harder. Well, what kind of perturbations would actually perturb directly these control parameters? And then I could go back and see, do they have a causal relationship with the signatures that I pretend have led me to look?
Can't you use your way? So let's assume that the TC, for example, will be variable. And then you model the dynamic and you basically see what will be the pattern that you observe and whether this pattern will be precise and reproducible.
Right, but I need to find a perturbation that makes the TC variable.
Oh, no, but you do it in the model. Just in the model to see whether it's a sufficient condition.
Well, I don't have a model. There's no model here. There is no model. That's important. This is all empirical. Model would be Newton, you know. This is empirical. I just looked at planets on the sky and boom, oh, there's relationships, but with transcription. You understand? I don't have a model, but now somebody could cook up a model. That's right. Somebody could cook up a model where they reconstruct all of this by having these three control parameters, where they reconstruct the embryo, where they find scaling, precision, and reproducibility. And I can lead you to a paper where they try to do this, and I can tell you it is damn difficult. Because now we have so many constraints, macroscopic and microscopic, it is very difficult, but it's good, right? That it's so difficult. Because maybe finally we get to some really universal model that tells us something that is good for anything we throw at it, any gene, any organism, whatnot. This is transcriptional regulation. Plants do it, right? Ideally, we have a model that plants fulfill as well. Yes?
The fact that the K is largest for yeast and smallest for humans at Drosophila, something somewhere in between, does that mean something in terms of developmental?
Right, good point, valid point. I don't want to go there because it would be purely speculative. to give you my personal opinion, I don't like that they don't fall on top of each other, even though I accept that it might be an important regulator across species. But because I disliked it so much, I moved across the ocean, opened a new lab, and started to work on a different model organism. And that's what I want to show you in the last 10 minutes, the beginning of it at least. Okay? Because everybody tells you, yeah, you work in flies, everything, especially in flies, and also, I don't like to take other labs' data as a validation or disvalidation of what we did here, because it's unfair. For that lab, for my lab, for anything, it's much better if you have something like I showed you on the previous slide with the data points, that you do it that you try doing this in the same lab, because every lab does things slightly differently. Okay, it's not a fair comparison. All right, and so going to a different model organism now, don't expect to see this stuff in the new model organism. This will take me another 10 to 15 years. Okay, but maybe we can start with the macroscopic constraints. So that was the initial quest. All right, so what did we do? We started to work with mammalian embryonic stem cell derived gastroloid. You might have heard about them. It's a mouthful of words, but basically these are multicellular aggregates that recapitulate key early developmental events in mammals, in this case in mouse, because we start them from mouse embryonic stem cells. These are basically organoids, right? If you take intestinal stem cells, you make intestinal organoids. If you take embryonic stem cells, you make embryoids. And we make a special class of embryoids, which are called gastroloids, because they gastrulate. Basically, they express genes that at similar stages of development in the mouse do gastrulation. That's roughly the upshot of what they are. And here you're basically looking at a patterning gene. This thing here is this band was a patterning gene. And one of the reasons why I use them is because the real reason from my perspective is just because they're roughly the same size than fly embryos, and I can use the same microscopy that I know how to do. All right, that was my main reason. But also because I wanted to go to a mammalian system, and because in a mammalian system to look at embryo development, it's very difficult to do this in a uterus, as you can imagine, if you want to come with a big machine. And so here you can grow them in a dish, recapitulate development somewhat synthetically in a dish, but you know, they still express genes. And if you look at gene regulation, which we did in the past hour, they should still be good. All right. All right, so the first thing you want to then see is, well, is this again a system that can be used for quantitative investigation? And the first thing we looked at, same as in the fly, is it reproducible? Okay, and so here we track gastroloid growth and elongation over time. This is one of the first movies we made. They look not very proper yet, but in any case, if you measure the diameter, you see this is several specimens on top of each other. They fall on top of each other, so it looks kind of the beginning of reproducibility here. And we can quantify the same results in fixed preparations where we get the statistics higher. Is ever going to come up? Here. So here we have a higher n, and you see for different time points, the size falls on top of each other, so it starts to look like it's reproducible. From these movies, or these trajectories here, you can extract an effective growth rate. Which seems to be largely conserved also across different initial conditions, initial sizes, etc. And even more surprisingly, it turns out that the total number of cells in these gastroloids scales almost perfectly with the initial number of cells. So here on the x-axis, you see the initial number of cells that you count to 300 and then put them in a dish, or you count to 100 and you put them in a dish, and you end up with 10,000. And what this graph here shows you is that if you start with twice as many cells, you get twice as many cells at the output as a function of time, independent of time, et cetera, et cetera. That's what the diagonal would give you. And the dashed line here is essentially our experimental error, the error in the growth rate and the error in counting N0, right? So it looks pretty good. And so already at the level of multicellular growth, we see signature of system level constraints. All right, this is not just randomly going this system. But of course, growth and axis elongation are tightly coupled to gene expression patterning. So the next question is, how reproducible are those? And so here you see SOX2 expression for 50 gastroloids. You can extract expression profile computational along this medial axis here, okay, that you construct computationally. And again, when we overlay individual profiles, you see that they all collapse. The variability peaks in the boundary region and remains low in the fully expressed domains at levels comparable that has been measured previously in fly embryos, in yeast, in C. elegans, and in bacteria. So again, something that gives us confidence that we can measure at similar levels, at least, than in these other species. And in the last panel down here, we did the same experiment 3 times, and again, the profiles collapse without any normalization on the y-axis, which means that cells reproducibly produce concentration in each position with just a few % variability, which is quite remarkable, because this is not an embryo. This is a self-organizing structure of cells that didn't evolve together. They were just come from a freezer. I throw them in a dish. The thing spontaneously self-organizes, elongates, and after 5 days, a cell at 35% medial axis length knows it needs to make 5.5 nanomolar of SOX2 and not 5.6. Kind of weird, right? This is not an embryo that had evolution to constrain it. Just to keep that as a thought. And again, this doesn't hold just for SOX2, but for all antibodies that we found in the system for this stage, which was 4. It's not that many, but it's a beginning, okay? So now the natural next question is, how precisely are boundaries positioned? There are 2 ways to estimate a boundary. The simplest is geometric. You can just ask here in this boundary, where is the half maximal point, and then look at this across many boundaries and construct a distribution. And the distribution has a positional error of roughly 2.5% of the length of the medial axis, which corresponds to roughly 40 micrometer. But cells don't measure position directly. They extract position by measuring concentration of genes. And so we can again estimate positional position by error propagating the variability from the gene expression profile. And then we get a function, as you see in the next panel. And you see that in the boundary region, it's very low. In the boundary region, it's very low. How low is it? Well, it turns out that this line here and this number here correspond to the size of a single cell, which is something that we measured independently. And again, this holds for all of the genes that we measure.
That's true only in the boundary region.
Yeah, that's only true in the boundary region. Like in the fly embryo. Well, in the fly embryo, it's a little, yeah. Let me take that back. And so even in this self-organized system, again, we see that patterning operates very reproducibly and near single cell precision. And if you grow gastroloids at different sizes, the patterning, you see again, very organized patterning. But now you can ask the scaling question again. Here for those 4 different genes, I show you when we grow them from different initial sizes, you get them at different lengths, final lengths. So it's different than with primaries where you have to go out and collect a big distribution and work with the distribution that nature gives you. Here you can generate the size distribution yourself. If you look in absolute units, they spread along the x-axis, but if you rescale by system size, again, they collapse, and again, with just 1 or 2 cell diameters. All right. And so, again, we see reproducibility, precision, and scaling in this new system. These are, again, the macroscopic constraints that we saw in the fly embryo. Now, the next step would be to go and look again at the microscopic transcription level. Okay. Here, of course, the important difference is that these properties emerge in a complete different context because this is self-organized and, you know, in a way synthetic. There's no maternal blueprint. There's no predefined axis. The axis defines itself. And there's no imposed morphogen sources that could help with any of the constraints. They just emerge spontaneously. All right, and this could actually mean that these features are not features of development, as we thought in the fly case, but that they are really features just of multicellular organization. As soon as you do something multicellular, well, these things come together. Yeah.
There's a recent work from Arjun Raj, where he did spatial transcriptomics on the gastroloids. And my sense was that the expression was much more random for many of the genes, but maybe it is later than what you're talking about here.
Maybe, but it's also not done with imaging. It's also not done with imaging.
I see. You mean like live imaging or?
Well, yeah, right. So I'm not sure to what extent.
Spaces of, like islands of expressing cells at a slightly later stage, and I'm just asking whether what you describe here is like important for the early boundaries formation, which is very precise, and then.
Yeah, there remains to be seen, right? But when did he look? At what stage did he look? Because this is a day.
I think they're very similar.
Yeah, this is a day 5. Yeah, exactly. It would be interesting to see. So basically, this is with antibody stainings and antibodies we got from different labs or companies because they were available. We now have developed SM fish in this system. We again see something very similar, but now, of course, we have the luxury to look at any genes. And it might be now useful to go back, take Arjun's data, take a few genes from there and see what happens. But for now, for the genes that we chose, we see the same thing. Like for a housekeeping gene, for instance, counting mRNA molecules, we get the same number of molecules in all cells. And these are low numbers. We're talking, 6 ± 2 in all cells. These are 10,000 cells, which is quite remarkable precision. Yes.
You mentioned multicellularity, but relative to what you showed us before in terms of those rates that are constant and so on, and you had data from this also. I don't understand why you think that multicellularity has, I understand that to show precision here, you need a multicellular system, but why do you think that multicellularity has any role in the emergence of
good point.
precision?
I don't know. It's an intuition. But
You show,
no, I didn't show anything.
No, but show, I mean, the data from the other lab show that the
Well,
same principle.
No, but sorry, but they're different, right? Those are the transcription,
Yes.
the transcription measurements,
But-
the transcription parameters. This is not precision or reproducibility.
No, I know, but
Right.
you assume.
I think for precision or reproducibility, you might need multicellularity, but also this remains to be seen. You're right.
But it's related to
But
the
it's
question
different.
of whether what
You-
you measure
right,
is
right, right.
sufficient or
Exactly.
necessary.
Right.
Here you are saying that it's
Absolutely.
probably not sufficient
Yes.
or necessary. You need multicellularity on top of it to achieve such precision.
Right. So when I said it's my intuition, with fish now, we can check. And so this gene that I just mentioned in gastroloids, 6 plus minus whatever 2 messages per cell. You make that same measurement in cell culture with the same gene, because in this system we can, twice as noisy. So there is a stabilizing event, or there is a stabilizing phenomenon that happens when you give cells a context, when you put them together with their friends, basically.
But if come back a little bit maybe to the question of Olivier, whether when you presented this layout, when you only assume you show transcription and no signaling, for me the multicellularity come with signaling. So
Yes.
So
yes.
I was wondering whether anyhow you would need to have signaling in order to get such precision.
Maybe. But again, that's another research direction. So I'm done. So basically, these are my take-home messages. I can just flash them. I don't need to go through the slide too much. I guess we want to move on. But basically, the one thing I want to basically take you away is that development might operate under these universal physical constraints that govern multicellular organization. It doesn't mean that we don't need molecular mechanisms, but it could mean that these constraints basically give us low-dimensional conditions within which the molecular mechanisms are allowed to operate. Okay, and so development is not just there's a genome and the genome does it, but there may be physical principles that are outside of it that basically give it constraints. And I'm not the first one to say this. We're going back to Darcy Thompson and Waddington and all of these people touring. They had the same kind of vision, but now we can actually measure this. Okay, I think that's what I wanted to get at. Of course, I want to close by thanking all the many people who made this work possible, if the slide ever comes up. I'm only listing the people whose work directly contributed, current and former team members, collaborators, and of course, special thanks to the current labs at Princeton and Pasteur and the funding source without which this work wouldn't be possible. And thank you for your long attention. I know I went way over time, and for all your good questions, and hopefully we can discuss more later. Thank you.